I think many of us have looked at these kinds of receptive field properties in the hippocampus and wondered where they come from. How come some neurons fire in some locations and some neurons fire in others? And it's not like we have an organ in our body that's telling us where we are, like we do for the pitch of sound we're hearing. And so these kinds of computations must come from the synapse. And I think that many of us have this kind of cartoon canonical model of synaptic function, where there's a set of presynaptic neurons here, and that activity flows over some synaptic weight, which is usually represented as a scalar. It's integrated and passed through some nonlinear operator in the postsynaptic neuron, and then we see the output. And usually us as the extracellular electrophysiologists then measure that kind of output. And so somehow place fields must come from these kinds of synaptic computations. And I think that a major goal of neuroscience, not just in the hippocampal world, but across neuroscience, is trying to figure out how is it that changes in the synapse relate to changes in neurodynamics and neural coding. That's been the focus of my career, and today I'm going to tell you about some of the little bits of headway we've made towards that kind of understanding. I think that an important first place to look at this kind of analysis is on fine timescale dynamics, because synapses operate on fine timescales. And so when you look at ensembles of neurons in the hippocampus, on these kinds of theta timescales, fine timescales, what you can observe is that some neurons seem to fire in close synchrony with one another and in opposition with other neurons. And so these have been called cell assemblies. And this kind of observation suggested that maybe what is going on is some kind of mutually inhibitory process, where those neurons that fire at let's get the laser pointer. Some neurons that are firing together at this moment are inhibiting these other set of neurons. And now these neurons fire, then the lateral inhibition kicks on, creating this kind of mutually exclusive pattern of activity. And so just updating this kind of cartoon model, maybe what's dictating when a neuron can fire and how it fires in response to different kind of stimuli is not just the pattern of excitatory inputs, but also the pattern of inhibitory inputs as well. I should say that this is actually a controversial proposition. But if it's true, then I think that it makes the problem a little bit more tractable, because these excitatory inhibitory synaptic connections we can actually estimate using the activity recorded extracellularly, and I'm going to show you how we do that. And so this is the kind of data that we record with silicon probes. It's voltage over time. Every trace here is from an electrode that's going dorsal to ventral in area CA1. These tick marks are just the spikes, and I just highlight this one event here where one neuron in black is fired just before another neuron in red. This is 1,000 such events. The black waveform is from an excitatory neuron, and the red is from an inhibitory neuron. And we can tell that just from the shapes of the waveforms and the firing rate. And I hope you can appreciate that there's this accumulation of firing of the inhibitory neuron just in the milliseconds after the excitatory waveform. We can quantify that kind of relationship using this kind of cross-correlation analysis, where the dashed line here shows the spike of the excitatory pyramidal neuron, and we can just accumulate how often do we see the inhibitory interneuron firing in the milliseconds before and after. And what we observe is this very strong peak, a high frequency delayed peak, and the delay is at the synaptic timescale, suggesting that maybe this kind of correlation is coming from a causal interaction, namely the synapse. People have been looking at these kinds of correlations since the 1960s and dreaming of reconstructing the anatomy. But the problem is the brain is very complicated. This synchrony could come from a true synapse, but we could be tricking ourselves. We could be recording from these two neurons, and they could be driven by some hidden third party, which is making neuron 1 fire at a phase delay from neuron 2. Or maybe it's going to be some kind of combination. And so the trick is, how do we make a causal statement just by looking at the correlation structure between these spike trains? And so what we needed to do is we needed to decouple the presynaptic pyramidal cell from all of these hidden third parties that could induce the kind of synchrony between the pairs of neurons that we might then infer as a causal interaction, but it's not. And we did this in a couple of ways. I think the most compelling was these experiments that I did with Dan English, where we glued a glass pipette to a silicon probe, and then we did juxtacellular stimulation of the presynaptic pyramidal cell and extracellular recording of the postsynaptic interneuron. And the strength of this manipulation is now that we can inject current into the presynaptic pyramidal cell and make it fire, and just that one neuron. And then we can record what is the response to the postsynaptic interneuron. Juxtacellular recordings are kind of a funny technique. It's half extracellular, half intracellular, and this is just what the spontaneous spikes look like of the presynaptic pyramidal cell. These spikes are created by the brain without the current injection. And now we can do the same kind of cross-correlation analysis with the extracellular recorded interneuron, and we can see these kinds of peaks, suggesting the causal interaction due to the synapse. Then we can inject current into the presynaptic pyramidal cell, thus decoupling the spikes from all of these hidden third parties that could possibly synchronize the pair of neurons. And we can make the same kind of correlation between the presynaptic pyramidal cell and the postsynaptic interneuron. And again, now we can see just for the evoked spikes, response in the postsynaptic interneuron, again, suggesting the existence of the synapse. We did these experiments 30 times. Every row here is a pair of pyramidal cell to interneuron. And you can see that some of these have strong spontaneous connections, some are weak. And what was very striking to us is for the evoked case as well, not only could we always see the peak, but also the size of the peak was very strongly correlated, showing that correlation over here. So not only can we estimate the existence of the synapse, but maybe we can get some estimations for the pairwise strength of the synapse through this kind of analysis as well. And so what we can get from doing this kind of analysis of the fine timescale spike-spike coupling is reconstructing kind of the pattern of recurrent inhibition in CA1. And shown in gray is the silicon probe that we're using to record neural activity. We can reconstruct the location of the pyramidal cells and triangles and the interneurons and circles relative to the shanks of the silicon probe. Here we just jitter a little bit around the shank just for the visualization. And then we can estimate what's the coupling strength between the pyramidal cells and the interneurons color-coded in red. I hope you can appreciate there's a lot of structure here. Most of the feedback inhibition is local. There's some very uneven connections here. Here's two interneurons side by side. For whatever reason, one of them receives inputs from almost all of the neighboring pyramidal cells, while the other does not. We don't know why that is. But of course, if I showed you these kinds of connectomics from electron microscopy, you would believe it more than this kind of correlation. We miss a lot. We can only detect the strong synapses. But what we can do that the electron microscopy folks cannot do is now we can look to see how these synaptic connections change over time. I think that most models of memory and neural computation rely on changes in the synaptic strength. And so the next challenge is trying to get a time-resolved estimate of synaptic coupling strength. And when we say change over time, and this is a whole conference about time, what do we mean? Time can exist on many different time scales. We could look to see over the millisecond time scale. For example, this would be presynaptic history or the ongoing assembly activations in the network. On the second to minute time scale, for example, if I'm sitting in my office or down in the laboratory, or learning a memory over the course of what we heard from Carrie Huffman today over the course of years even. And I think that mostly these long-term scales is what people are interested in learning a memory field. But just to start, we can make some progress, I think, even looking at these short time scales too. I debated as to whether to include this, but I think because we have this other session about sequence generation, I think that kind of the short-term plasticity could be a powerful theoretical mechanism for allowing sequences of neural activity to unfold. And so I include a little discussion here. And it's well known that the excitatory to inhibitory connections in the many synapses across the brain show short-term depression and facilitation, where if you see a spike train of presynaptic inputs, you see that the postsynaptic response sometimes gets weaker, and that's known as synaptic depression, and sometimes gets stronger, and that's known as synaptic facilitation. And so can we see the hints of synaptic facilitation and depression just by looking at our spike-spike analysis, like I showed you before? And indeed, I think we can. Here's how we did the analysis. What we did is we conditioned the presynaptic spike on whether another presynaptic spike had happened previously at different timescales. So in this case, we see a burst where two presynaptic spikes happen at 10 milliseconds. And then we say, what is the potency of the drive of the second spike in the burst in driving a postsynaptic interneuron? What's the probability of observing a postsynaptic spike? And then we can plot that. And then we can pull out what is the interspike interval for the presynaptic spike, and always asking after the second action potential, what is the likelihood of seeing the postsynaptic neuron fire? And I hope that you can appreciate in this case, it looks like this is synapse is acting as a high pass filter, where if the presynaptic neuron is firing at a high firing rate, which is a low interspike interval, the synapse is relatively depressed. While at the low firing rate, we see very strong spike transmission between this pre and postsynaptic neuron. This is considered the rule between the drive to these fast spiking interneurons, and almost all of these are fast spiking interneurons. But indeed, we see other cases where we see what looks like synaptic facilitation, where it's a high pass filter, where the firing at a high firing rate to the presynaptic neuron seems to potentiate the activity of the postsynaptic firing. And indeed, these two different filters are derived from the same presynaptic spike train. So two different postsynaptic interneurons can read out different patterns from the same presynaptic spike train. And I think that often in the interneuron literature, it's easy to say that these kind of patterns are due to there being different kinds of interneuron types, but that's not the case either. We can look to see for the exact same postsynaptic interneuron, different kinds of synaptic filters, where some look like synaptic depression, some look like synaptic facilitation, and some look like a combination, and actually many look like this, where there's some kind of bandpass filter with an ideal frequency of communication between the pairs of neurons. Okay, so just in kind of building up for complicating this classic view of synaptic computation, I think it's probably been recognized for a long time, but it's nice to see a reminder that these synapses are not scalars. It's frequency dependent, and we're operating fundamentally in the frequency domain. And I think that in terms of models of sequence generation, if the role of this kind of inhibition is to have competition in CA1, then you could imagine that as the drive from a pyramidal cell to its neighboring interneuron wears off because of synaptic depression, this could now allow another set of pyramidal cells to fire. And the kind of bump of activity could move around sequentially from one population of pyramidal cells to the other at the time constant dictated by short-term depression. That's just an idea for a model. Okay, so how do these synaptic couplings change over time? We kind of talked about these short time scales in the form of short-term plasticity. I'm going to skip over a discussion of changes in behavior because we only have 20 minutes and go straight to trying to estimate over long time scales, for example, with learning and plasticity and neural coding. And I was very inspired by the set of experiments done on Janelia Farm and Jeff McKee's lab and others, where they were able to turn silent cells into place cells just by injecting a large amount of current into this intracellular into single neurons. And here what they had the animals doing is they had the animal head fixed running on a track. This is the activity over trials. This is position. You can see at the beginning, this neuron didn't fire. They induced these plateau potentials by injecting a large amount of neuron into single neurons, and now this neuron fires in this location trial after trial, suggesting that there's somehow this neuron has been married to this location on the track. We had many questions about this. For example, we know that this is not the only neuron in the brain, and there's probably other neurons that are firing in this location. So how does this neuron become incorporated within the overall ensemble of cells that are coding for similar places? Could you do this experiment forever and just have 1 million neurons coding for a single location? Probably not. So we wanted to kind of scale these experiments up and rather than controlling single neurons at a time with the intracellular glass pipette, use an optogenetic approach. And so here, in collaboration with Yusuf Yoon and the micro-LED team at the University of Michigan, we recorded neurons with these micro-LED silicon probes, where there's LEDs microfabricated amongst the recording sites. These are tiny, 10 by 15 microns, and we can depolarize very small numbers of neurons while recording their activity. And so what did we do? We had the animal freely behaving, running back and forth on a linear track. We had an infrared detector placed at a random location on the track. We used that to deliver this kind of envelope of light power to depolarize subsets of neurons. And so what does the data look like? This is data from a single neuron. It's positioned on the x-axis, trials on the y-axis. We did a morning session and the afternoon session to test for the stability of the place fields. This is a 2 to 4 hour break here. And this neuron, you can see, is a very stable, robust place field, fires almost every trial. The blue shading here shows where we turn on our light. And you can see that in this case, we can drive the neuron and it continues firing in this location. In this case, we did the stimulation twice, and this newly induced place field continues into the afternoon. So it's stable over the course of hours. So if every neuron looked like this, I'd say that we have a nice model using optogenetic stimulation rather than cellular current injection for the induction of kind of this finger of God experiment, where I can make the neuron fire whenever I want, however I want it to do. But this is not what we saw. We saw many other funny things too. For example, here is a neuron that has a place field. We stimulate another place field remaps to some other location on the track. We also saw cases where now this neuron was not stimulated. This was recorded on a neighboring shank. You can see that we turn on the light and the neuron does not fire, but now we also see a new place field emerging, which is somewhat stable even over the course of hours. And so what is going on here? I just want to convince you that our stimulation, we did not see remapping to the stimulation site. Here's showing the firing rate at the stimulation site in blue and out of the stimulation site in red over trials. We stimulate normally for 5 trials here. You can see this massive increase in the firing rate, and there's no hint even that there's an increase in the firing rate at the stimulation site afterwards. This is all of the neurons that showed remapping. This is their place fields before we stimulated. This is during stimulation and afterwards. You can see I lined up all of the place fields to the stimulation site, so we get a strong drive here. And they're sorted according to their post-stimulation place field location. So they could remap anywhere on the track. This is now our afternoon session, and you can just see qualitatively, and it actually turns out quantitatively as well, that this pattern immediately after the stimulation is more similar to the afternoon session as it was to the pre-stimulation session, even though these are only separated by about 10 minutes rather than 1 to 4 hours. We're battling the fact that this analysis that the place fields can drift all of the time. It's a very modest stimulation that we give, and so we quantify the remapping in several different ways. Here's one of the ways that we did that is we built a template of what the place field looked like prior to stimulation, and we correlated that template with the activity trial by trial. And you can see in this case, the pre-stimulation template correlation is high, and then it drops off around the time of the stimulation when the cell remaps. We can flip the analysis. We can build a post-stimulation template, correlate this trial by trial, and now we see the converse pattern where the correlations are low and then they increase. So the difference between these pre and post-stimulation template correlations determines the degree of remapping, and where they switch over, this is telling us when the remapping occurred. And so for the stimulated chunks, we saw indeed that there was a big difference here between the pre-stimulation template and post-stimulation template correlations. Interestingly, this post-stimulation template correlation begins to increase even prior to stimulation, suggesting that somehow those patterns are already in the network. I'll talk about that a little bit later. But we didn't only observe this on the stimulated chunk, but also on the non-stimulated chunk. And both of these were significantly more than what we observed in control cases where we never turned on the light at all. Interestingly, we did these experiments both in CA3 and the dentate gyrus, and in both cases, the place fields were rock solid. So this kind of plasticity that we observed in CA1 looks like it's fundamentally different from CA3 and the dentate. We wanted to dig in a little deeper and say, where did these place fields come from? In this case, what we did is we took all of the neurons that we mapped and we lined them up to the peak position of where the new place field would be. And then when we look back in trials, we see that even before the stimulation, we see this little hint of firing. It would never be quantified as a frank place field, but it's not nothing, suggesting that there's some pre-existing drive at this location. And the kind of remapping that we unleash in this case is more like what's been discussed as rate remapping. And so we wanted to ask ourselves, well, maybe what's happening with our stimulation here, maybe why we're able to see remapping even for neurons that are not being depolarized with our light, is because we're causing a reorganization of feedback inhibition or recurrent inhibition. And so we wanted to ask ourselves, do these new place fields emerge because there's some sub-threshold existing field that's now disinhibited. And so what we needed is a time-resolved measure of the synaptic drive from the pyramidal cells to the interneurons. And so what we did is we built a statistical model, and you can read our neuron paper to try to dive into the details of that. But we can now estimate what is the instantaneous drive of the pyramidal cell to the interneuron over minutes and hours. And I hope that you can appreciate that there's a huge amount of fluctuation here, likely due to, in part, to neuromodulators. And you can see that when the animal's in the home cage, this pair has a significantly weaker drive than when the animal's on the track. And you can see that both as the data and in the model in the cross correlations. This is now an ensemble of synaptic coupling strengths over time. And you can see that there's a huge amount of covariant structure here, where some of the synapses like to be strong together and others like to be weak at the same time. And it looks like that there's this kind of mode shift when the animal goes onto the track. If I wanted to show you quantify this matrix here, I don't need to have the full 60 rows, which is every pair is a pyramidal interneuron pair. I could have a kind of lower dimensional representation of this matrix here. And so as people talk about cell assemblies, we can talk about synapse ensembles, which are kind of what are those different synaptic modes, which are describing the set of synapses that rise and fall together. And so this is what we did. We described those kinds of assemblies of synapses. And what we observed is that some of them were expressed early, and those tapered off towards the end of the session. And some of those were expressed late, and those were weak at the beginning of the session, suggesting that there's a different kind of graph describing recurrent inhibition in CA1 before stimulation and after stimulation. And we can quantify how much this graph changes before and after our stimulation in the stimulation case versus the CA1 control and the dentate gyrus. And what we observe here, and this is this axis just showing how big of a change we see in these kinds of synaptic assemblies. Negative shows more change. That when we stimulate, we get much larger rearrangement of the synaptic strength. And CA1 when we stimulate as compared to the no stimulation sessions as compared to CA3 and the dentate when we did not observe the place field remapping in vivo. And so I think that this, for me, has really inspired this more complex view of synaptic computation, where maybe the synaptic strength is not really a scalar, but it's a function of the ongoing activity in the network. So the conclusions here, it looks like that we can identify synaptic connectivity just by looking at these spike-spike relationships and even some of the short-term dynamics, like I showed you here with this synaptic depression. There's a focal optogenetic stimulation of induced place field remapping in C1, less so in CA3, the dentate gyrus. And this looked like it also correlated with changes in the coupling strength between excitatory cells and inhibitory cells, suggesting a model where reorganization of lateral inhibition could be what was driving the remapping, specifically in those non-stimulating neurons. I just want to say in my last minute here, some of the work that we've been doing in the lab. So we want to dive into some more of the plasticity rules between excitatory cells and inhibitory cells. So what we've done is we express crimson under the PV promoter and channel rhodopsin under the kinase promoter. And then we stimulate with red and blue light to control the activity of each of the two different populations of neurons. And what we can see is when we give theta burst stimulation pairing the two, is we can get long-term changes in the drive of both inhibitory neurons and excitatory neurons, suggesting that indeed these kinds of synapses are plastic in the brain in vivo. Here's just two control cases showing that some of the neurons show stable responses to light. Okay, so the implications we talked about. For me, this means that there's an important potential for memory in a region without strong excitatory connections. Synaptic coupling is likely dynamic. I'd like to thank Dan English doing the extracellular experiments. Praveen Padel did the recordings in the lab here at UNM. And this is my lab shown on the right. And please, if you're interested in this kind of work, come join me at UNM. I'm recruiting all the time. So no chicken sounds, though I know I'm over. Thank you kindly, everybody.